Chapter 8, Section 1, some basic definitions and conventions. This section, should actually be a repeat. Of intro cell and cell biology, genetics. So it shouldn't be too much here that is new to you. This idea of a gene. A gene is the blueprint on how to make one polypeptide. Okay? That's it. For all intents and purposes, that's it. Now, I am being a little facetious there. It's not just for making a protein. Sometimes these genes are just for making RNA. tRNA, ribosomal RNA. But predominantly the genes are for proteins. The instructions on how to make an enzyme, how to make a structural protein. Said structural protein will have some role in that cell. When it comes to the RNA, like I just said in the last slide, there's going to be ribosomal RNAs, tRNAs. Of course, there's the messenger RNA. It's what takes the blueprint information from the chromosome to the ribosome. A new area of study we're looking into, what's referred to as non-coding RNAs. These non-coding RNAs, you'll also see them referred to as siRNAs, silencing RNAs. Their job is not to code for anything. Their job is not to build anything or be responsible for aiding in building anything. Their job is regulation. Regulating transcription. Regulating translation. These small RNAs will interact with DNA sequences in the chromosome, interfere with transcription factors from binding, interfere with polymerases from binding. Some of them also interact with the messenger RNA itself. Make it so that the messenger RNA can't get in the correct shape, because it turns out messenger RNA is not, a straight line like it's always drawn in textbooks. It will form little hairpin loops and stuff to help stabilize it. Well, these little silencing RNAs can bind to that, keep those little hairpin loops from forming, so that the RNA is less stable. And as we're going to see in this chapter, RNA is inherently unstable and will start to fall apart all on its own. All of the nucleotides have three components, okay? A nitrogenous base, which is going to be up here like a flag, standing up. The sugar, it's based on pentose. This hydroxyl here is highlighted because it's the presence or absence of that hydroxyl. Presence, RNA. Absence, DNA. Ribonucleic acids. Deoxyribose. Loss of the hydroxyl. DNA. So there's the pentose. And then up here, attached to the 5' carbon, is the phosphate group. All three of these together, 1, 2, 3, and you have the nucleotide. Some places in the cell, having a different role than being part of a DNA or RNA chain, you're going to end up with some nucleosides, where it's just the nitrogenous ring and the sugar. They fulfill different roles. They will be attached. They will be doing other things. Now, when you look here at the pyrimidine and the purine rings, they are called nitrogenous bases because if you look here, this is not a carbon-carbon chain. Yes, there's carbon-carbon bonds in here, but interspersed within are nitrogens, hence the nitrogenous base. When you look at these nucleotides, you're going to see a specific bond mentioned. N-beta glycosyl bond. Okay? from the 1' carbon of the sugar, it's in the beta conformation. If you think back to the previous chapter when we were talking polysaccharides, at the 1' carbon, whether the hydroxyl is pointing down, pointing up, compared to the plane of the sugar, pointing up alpha, pointing down beta, or beta alpha. Well, there you go. That's the beta. The N, nitrogen, beta, glycosyl. Okay, this is telling you who's joined. So it's a nitrogen to carbon interaction here. It's formed by dehydration or condensation, tomato reaction. Remove the water, and there you go. The 5' N, you're going to get a phosphate group that is attached to the 5' carbon via an ester linkage. That means it's going to be a phosphate, P, bound to an O, bound to a C. Ester linkage. Two types of these nitrogenous bases, okay? Double rings, single rings. The double rings, adenine, guanine. The single rings, the pyrimidines, cytosine, thymine, uracil. A, G, C, T, U. Okay? A, G, C, and T are DNA. A, G, C, and U, RNA. Notice, yes, A, G, C is found in all RNA, DNA. It's the T and the U that are interchangeable, or I'm sorry, not interchangeable, will be found with one or the other. T goes to DNA, U goes to RNA. It's part of one of these things that besides just the oxy-deoxy of the sugar, the pentose, whether it's a T, whether it's a U, allows the cells and the enzymes to recognize whether this nucleic acid string is DNA or RNA. Certain enzymes can only react with certain sequences. AGCT sequence, DNA. AGCU sequence, RNA. So you look here, purines, pyrimidines, who, the what. You'll notice whether it's a nucleoside, nucleotide, adenosine, adenylate, guanosine, guanylate. Remember, this is the sugar and the nitrogenous ring only. This is all three, nitrogenous base, the sugar, and the phosphate group. Now, this sugar, this pentose, it's a 5-sided sugar molecule. The chemist will recognize this as aldehyde. 1, 2, 3, 4, 5 carbons. Carbonyl up here at C1. C2 has that, or C5 has the hydroxyl. This is the structure you would recognize. This is what biologists would recognize, because remember, we draw everything as though what it would look like in an aqueous solution. And as we talked about last chapter, with the sugars that are going to be in an aqueous solution at pH 7-ish are going to do this cyclic ring. This is more stable in an aqueous solution than this structure here. But as we saw with the glucoses when we were talking the polysaccharides, it's not a flat structure. The artists tried to mimic this here by showing the wedge, thick line here, that this edge of the sugar has a little bit of a tilt up. Okay, it tilts up right there, as you can see here. Think back. Last chapter we're talking polysaccharides. Instead of the nice six-sided ring, it's actually distorted and has a slight twist to it. Same thing here. But we're still going to draw it like one of these two. Okay? If you're drawing it as, part of a nucleotide or part of a DNA or an RNA strand, this is how the sugar should look. This is a little too much for us to sit there and play with. So we're not going to worry about that. These deoxyribonucleic acids, what we refer to as DNA nucleotides. The correct terminology for them is actually deoxyribonucleoside 5' monophosphate. But you know what? That's just a little too much to say. I mean, when you look down here, deoxyadenolate, deoxyguanolate, deoxythymolate, deoxycytolate. You're going to hear most biologists going to refer to this as adenosine, guanine, thymine, cytosine. The truly more correct term for the name for these, deoxyadenosine 5 monophosphate. Yeah, that just doesn't work. Sometimes in some metabolic pathways, metabolic reactions, what was a triphosphate, ATP, GTP, TTP, CTP, the two terminal phosphates will be cleaved and you end up with the monophosphate. So you'll see it sometimes as AMP. Some chemistry journals, chemistry textbooks, some primary literature coming out of chemistry-based journals will refer to it as DAMP, DGMP. If you're looking mostly at a biological publication, they're just going to write AMP, GMP, TMP. Same thing with the RNA bases. Again, how do I know it's RNA? Well, right there, hydroxyl, 2' hydroxyl. Ribonucleoside, 5 monophosphate. Adenosine, 5 monophosphate. Guanosine, monophosphate. Here it's just AMP, GMP, UMP, and so on. When you look at just the nitrogenous bases, the purine and pyrimidine bases, what we find is that sometimes there's going to be an extra functional group attached, specifically a methyl group. We call this methylation or methylated form of the DNA bases. Its base, the nitrogenous rings base, the nitrogenous base, is similar to what we've seen on the past few slides. It's just there's going to be a methyl group will be added somewhere on there. Purpose of these methyl groups is to slightly alter how these nitrogenous rings will interact with other nitrogenous rings. It's going to slightly change the DNA double helix. It's going to change what we're going to see in the next section as we're talking the structure of the double helix. It's going to put slight bulges, slight changes in how it does the double helix. It's going to make it tighter, or it's going to make it looser. It's going to actually, instead of being the right-hand twist, it's going to make it a left-hand twist. Weird things like that. We also see that this methylation is a mechanism that animals have utilized, evolved to utilize, that aids in the choosing of one chromosome over another. Because think about it, you have two chromosomes, one for mommy, one for daddy, for chromosome 1, 2, 3, 23 pairs. Remember, one for mommy, one from daddy. Well, for some genes, two pair or a pair of that gene is one too many. You only need one. One is going to act as give us enough mRNA. It's going to be copied enough, give us enough mRNA that we're going to make enough of whatever that protein, that RNA strand is going to be that we don't need anymore. Having 2 open and available could be, too much. So, methylate. The methylation will mean one will be, chosen, preferentially over the other. They're both there, but, one's going to be more often than the other. So instead of being equal, 50-50, it's now going to be more this one and maybe once in a while this one, but mostly over here. It's our way of getting around it. We also find that this 5' phosphate is not always the way everything's going to look. Some of the times it could be shifted to the 2 or the 3' carbon. Why? Well, if you look here, adenosine 3 monophosphate. Okay? Adenosine 3 monophosphate is what happens when you have an RNA strand and an enzyme comes and cleaves, breaks, hydrolyzes. Okay? You don't want RNA, messenger RNA specifically, hanging around too often. It's got a short lifespan. You want it there, you want to have it read by the ribosomes a couple of times, and then gone. So enzymes cut. When they go to cut, they don't cut right here, so you end up with the true nucleotide, the nitrogenous base, the sugar, the 5' phosphate. No, what they're going to do is they're going to cut in such a way, they're cutting right here. They're going to cut, boom, right there. So this phosphate group is still attached to the preceding, as you see right here. Still attached. The cut right there means that that phosphate group is now attached to whatever preceded it. So it's a remnant. It's an artifact. But it's okay. It's not like this is all waste. What'll happen is that enzymes will come through and will remove this phosphate group. You now end up with a nucleoside. Another enzyme will come and strip the terminal phosphate off ATP, or just grab an inorganic phosphate that's floating around, add it right there. Boom. You now have the nucleotide reformed. What I was talking about on the previous, you have this RNA strand here. Instead of cutting like you would think right there, giving you one entire nucleotide, another nucleotide, what happens is that enzyme cuts right there, cuts right there. Nitrogenous base, sugar, 3' phosphate, completely backwards. But when you're looking at this DNA strand or this RNA strand, you need to notice a few things. You need to be cognizant of a few things. Notice right here, 5', 3'. Just as when we were talking polypeptides, we had to be cognizant of the amino terminus, carboxy terminus, so that way we had orientation down. So that when we're numbering amino acids and we say there was a change at amino acid 87 from alanine to leucine, you knew starting at the amino terminal end, you count 87 and there's where the swap occurred. Same thing here. Instead of an amino terminus, carboxy terminus, we have a 5', we have a 3'. 5' is going to be the first nucleotide, and it's going to have that phosphate group stuck out there. The 3' terminus, that nucleotide is just going to have the 3' hydroxyl sticking out. 3', 5'. Discussing this is going to be very important, especially when you come to like the homeworks. And it's going to be talking, hey, looking at this DNA strand, what is the complementary base pair? Well, you're going to sit there and you're going to have to sit there and say, okay, here's the 5, here's the 3. The complementary strand is going to be the reverse. 3 up here, 5 down here. And that's how you're going to have to discuss things. Same with the RNA. Has a 5' end, has a 3' end. Why is this also important? Well, this phosphodiester linkage right here. Polymerase, DNA polymerase or RNA polymerase, is going to take the incoming nucleotide, and there's going to be a condensation reaction right there. This hydroxyl that was terminal at that given instant is going to be removed, and now we're going to get this phosphate group being attached right there. Phosphate, 5' carbon, 4' carbon, 3' carbon, phosphate, 5' carbon, 3', 4', 3', phosphate. There is our sugar phosphate backbone. What we see is that RNA, by its very nature, is unstable. Okay? Like I said a few slides ago, we want messenger RNA here just long enough for it to be read by a couple of ribosomes so that the polypeptide can be made, and then we want it to go away. We want it to break down. Yes, there's going to be enzymes to help it, but as we see here, there's also going to be some electron attacks, some nucleophile attacks here. The electrons are going to shift so that this gets shifted around and things get reformed. It just falls apart. Hey, look, there's one of those cyclic nucleotides we talked about a couple ago. And it happens, notice there's no mention of enzyme. This will happen on its own, spontaneously. Hydroxyl attacks here, that hydrogen pulled off, that bond is going to shift, boom. So now here, instead of being oxygen to hydrogen, it's going to be oxygen to this phosphate group. This here, this bond here, is going to shift over, and with a nice hydrogen there, you now have a hydroxyl sticking out. So this oxygen and that hydroxyl was part of this phosphate group. It's now being supplanted by an attack from this oxygen. So notice the double bond here is now shifted down. Boom, boom. This oxygen remains stuck to the 5' carbon and now becomes a hydroxyl. This is spontaneous in RNA. Less so in DNA. Due to its double-stranded nature and whatnot, DNA is a little more stable. Okay? RNA, very short shelf life, measured in minutes, hours at best. DNA, heck, we can still isolate some DNA from, 3,000-4,000-year-old mummies. Woolly mammoths that have been frozen for 50,000 years. RNA, yeah, not so much. Now, I know you're thinking, okay, but messenger RNA falls apart really quickly. Okay, I get that, but what about the other RNAs? The ribosomal RNA, the tRNA? Well, as we're going to see in the next sections in this chapter, the tRNA has so many stem loops where it bends back on itself that it forms a pseudo double-stranded RNA. It aids in stabilization. The ribosomal does that, but it also has some small polypeptide fragments that are going to be there and aid in stabilization of the structure. You're going to see when you're reading certain textbooks, when you're reading certain chemical or biochemical journals that they will use this as a means for displaying a nucleotide sequence. Here's the 5' end, 3' end. 5' has the phosphate, 3' has the hydroxyl. And they will go through phosphate group, 5' carbon, sugar, nitrogenous ring. Here's where the 3' phosphate, that phosphodiester bond, phosphate, 5' sugar, nitrogenous ring, 3' to phosphodiester, phosphate, and so on. Boom, boom, boom. This is not something we do in biology. Okay, I've never seen this in a biological paper. We'll not basically just list out AGCTA, and we're just going to assume all of the rest of this. Just like here where they're showing, hey, look, P, saying this is the phosphate, ACGTA hydroxyl. What you're going to see more often than not, if you're looking, you'll see these three in, like I said, chemistry, biochemical. In biology papers, you will just see A, C, G, T, whoops, sorry, T, I'm trying to do this with the mouse, A, not quite so spaced out, and then it'll say 3'. This, bio, these, chem, okay, or more correctly, biochem papers, okay. You also see the terms tossed around, ligonucleotide and polynucleotide. If it's less than 50 nucleotides in length, we call that an oligonucleotide. These will be anything from 1 to maybe 3, 4 dozen at the most nucleotide sequences. They have their own jobs. They have their own things, whether it's DNA or RNA. But then you're also going to see much, much longer. How much longer? I don't know. Hundreds, thousands, billions of nucleotides. Yeah, that's polynucleotide. When you look at the structure of these nitrogenous bases, okay, they're not flat. Remember the sugars had a little curl at the end. Well, these guys aren't going to be flat either. They're going to have little twists, little tweaks of their own. Pyrimidines are going to be more planar, not so many twists, tweaks, bends, and curls. The purines are going to have a slight pucker effect, so it's going to be more of this type of thing. Whether it's a plane, whether it's a pucker, whether it's got a slight twist, a tweak to it, due to the methylation and whatnot, it's all for bringing more of the hydrogens and the hydroxyls into close approximation so that there can be complementary base pairing, A to T, G to C. And it turns out that it's not only methylation or things like that that's going to occur to the nitrogenous rings, but there's also some what they refer to as tautomerization, slight changes in the ring. Predominantly lactam ring, slight changes in the environment. Instead of the lactam ring, you get a lactim, double lactim. What's occurring here? Our carbonyl becomes a hydroxyl. What was the double bond shifts? Now it's carbon-nitrogen double bonds. Boom, lactim ring. Again, you see this double bond here shifts. Boom, carbon-nitrogen double bond. Hydroxyl left out there. And it just goes back and forth, until it's in a polynucleotide that is base pairing. bent back around, and you get that hairpin and stuff will stabilize that. When it's out on its own, free floating in the cytoplasm, you get this shift back and forth. We also are trying to come up with new ways to examine what nucleotides are present in a sample. What nucleotides are present free, what nucleotides are present as they are joined into a sequence, an oligonucleotide, a polynucleotide, whatnot. Turns out the nucleotides, all of them, have an absorbance spectrum of their own. We're still trying to figure out ways where we can utilize this to sit there and study cellular contents. But if you watch them and you look, you will see that the peaks shift, well, just don't shift enough for us to be able to sit there and run it through a spec and say, oh, look, that peaks this, that peaks that, that's that. But we're getting better. We're getting better at this as a means of not only knowing what's in the cell, but hopefully at some point in time being able to use something similar to this, with the right machine and the right programming to be able to sequence a DNA strand just based on, where the peaks are and in what order. Nucleotides on their own, not very hydrophilic. Okay, the nucleotides relatively insoluble, which is quite different than what we talked about in lab a few weeks ago when we were using those spin columns, remember, to try to isolate the plasmid DNA. I told you the plasmids, that DNA, was soluble in water, insoluble in alcohol. And we were using that because the proteins that we were trying to get away, extract the DNA from, were the opposite. Soluble in alcohol, insoluble in water. Turns out nucleotides on their own, not so soluble. DNA, RNA, get the oligonucleotide or a polynucleotide, changes the structure, changes everything about it, so it now becomes soluble. And it all comes to this. Base pairing, as I mentioned before, A to T, or A to U if you're talking RNA in the self base pairing, G to C. The two people you see here, Watson and Crick, yeah, I know, people automatically want to boo and hiss. Turns out, in their private lives, yeah, the guys weren't that great, okay? I don't care, all right? They were geniuses. They figured this out. They did, what scientists does. They take in information from wherever they got it from, and they used it. They used it to figure out the true structure, this complementary base pairing thing, and they also figured out 2 and 3. Individually, hydrogen bonds are extremely weak, easy to make, easy to break, but they're cumulative in effect. You take a chromosome of yours that has hundreds of millions of nucleotides base pairing together, as you see here, 2, 3, so that's 100 million nucleotide base pairs, 200 to 300 hydrogen bonds. Yeah, that's going to start to hold stuff together, but not so tightly that it can't be pulled apart. So yeah, they figured this out. They won the Nobel Prize, not just for figuring out here's the two complementary, the two complementary strands that are anti-parallel. Here's how the AT and the GC base pairs, but they also figured out here's how the semi-conservative DNA replication works, and quite a few other things. So yeah, I'm not going to sit there and begrudge them their Nobel Prizes. It's just as human beings, they were a little bit less than.